Hello, today we'll talk about recombinant DNA. At first, we need to know what's recombinant DNA. It's combination of DNA of two different species to add new traits or improve some characteristics in the species for medical or scientific purposes, like combining the DNA of human to the DNA of prokaryotes or bacteria. Here we have an example, making insulin needed by human, so they get human insulin gene and combine it to DNA of bacteria, so it makes rapid reproduction of new bacteria with human insulin DNA. Here are two pictures that show recombinant DNA. The first picture shows a DNA of human. It's cut by restriction enzyme. Then the plasmid in bacteria is being opened, and they combined the DNA from human to the plasmid, which is the circular DNA of bacteria. And in the second photo, it shows also the recombinant DNA. Now we will talk about steps of making recombinant DNA. The first step is using restriction enzymes, which are enzymes that cut DNA from specific sites in DNA and make parts called DNA fragments. These sites in which the DNA is cut from form sticky ends. Restriction enzymes are also called molecular scissors, and there are thousands of restriction enzymes. Sticky ends is the place where DNA from human joins the other DNA from prokaryote or bacteria. Here are two pictures that show how restriction enzymes cut the DNA. In the first picture, we see the restriction enzyme cut the DNA from specific sites and forms sticky ends. Then the DNA is recombined from the human DNA and the DNA of prokaryotes or bacteria. Here is step 2 of making recombinant DNA. Polymerase chain reaction, or PCR. It's a technique used to make millions of copies of DNA within few hours. It works by 3 steps. Step 1 is denaturing, and it's heating the DNA to separate the DNA strands. Step 2, it's annealing, and it's lowering temperature, so short DNA fragments called primers attached to the gene that is needed to recombine. Step 3 is extension, and it's a process in which they add enzyme exactly like DNA polymerase called the Taq polymerase, and it builds new strand. Here are two pictures that show PCR. The first step is denaturing in the first picture, and the two DNA strands are being separated by heating, then annealing, then extension. In the second picture, also denature or heating of the DNA strands so they can separate in 2 strands, then anneal primers to their complementary sequence, then synthesis of new strands with Taq polymerase. The last step and step 3 of making recombinant DNA, it involves combining the DNA that is made from PCR to the DNA of the species. Step 1 is removing genes needed from humans by restriction enzymes. Step 2, opening DNA of bacteria-like plasmids. Step 3, connecting DNA of human to the plasmid. After that, the bacteria start reproducing rapidly. Here is a picture of how step 3 is made. First, they got the DNA of human by restriction enzymes. Then they got the plasmid, which is the DNA of bacteria, and they open the plasmid and stick the DNA of human to the plasmid. Then they insert it again to the bacteria. Then this bacteria started to reproduce rapidly. Here are some important points. In making insulin from bacteria, they remove a circular DNA called plasmid because it's easy to move in or out of the cell. Scientists choose bacteria specifically because it divides quickly. Aims or purposes of making recombinant DNA. The first is making insulin from human gene by bacteria, and this is what we talked about. The second is medicine or vaccines. The third, making disease-resistant crops. The fourth, higher vegetation reproduction. The fifth, adding more vitamins to food. Thank you.